Welcome to this ECHA Science Seminar, which is today dedicated to new approach methods for adult and developmental neurotoxicity. So before we start, a brief disclaimer. So this seminar will be recorded and will be published later on the ECHA website. We want to have the seminar live and interactive, so use the chat anytime if you have any questions. And you can raise your hand and use the microphone during the Q&A session. ECHA does not endorse any of the tools or opinions presented today, and we will circulate a survey just after this seminar to collect your feedback for further improvement. A brief reminder of the ECHA Science seminars and the objective of those seminars. So the idea is to have presentation of scientific content that can be dedicated to human health or environmental topics. And the objective is to discuss the science, but also the regulatory relevance and the regulatory uptake of the result of those presentations that we can use that are relevant within our mandates. And finally, as I just mentioned, those seminars will be recorded and will be published on our new ECHA Science webpage, and more specifically under the Knowledge Hub page. So today the seminar is dedicated to neurotoxicity. So a brief background. So as you know, currently there is no hazard class under the classification, labeling and packaging regulation, the CLP regulation. The adult neurotoxicity is addressed under a specific target organ toxicity endpoint after single or repeated exposure, and the developmental neurotoxicity is addressed under reproductive developmental toxicity. As you know, we need to move away of animal testing, so we need to develop new approach methods, NAMs, and we are facing different challenges. Like, for example, how can those NAMs differentiate between INT, so adult neurotoxicity, and DNT, developmental neurotoxicity? How could those NAMs potentially fit in the current CLP hazard classes? Or how could they potentially support any future CLP paradigm for neurotoxicity? Can NAMs delineate between INT and DNT is the topic of today's seminar. This is part of a PARC project, so the Partnership for Assessment of Risk from Chemicals. The speakers of today will present the result of the PARC project, and they will also present the follow-up potential project that they would like to submit on the PARC. And so now I will warmly welcome the three speakers. So we have the pleasure to welcome Professor Dr. Fritsche, which will be the first speaker. She is the director of the Swiss Centre for Applied Human Toxicology in Basel, Switzerland. She has 20 years of experience in toxicology and 15 years in developing NAMs for neurotoxicity assessment. She has been involved in a lot of projects dealing with neurotoxicity, together with EFSA and US EPA. She is involved in a lot of European projects dealing with this topic, and she is also a member of the OECD DNT expert group. We also have the pleasure to welcome Dr. Jonathan Blum, which is working in the same institution as Dr. Fritsche. During his PhD, he has worked on advancement of NAMs and was one of the main contributors to the establishment of the DNT in vitro test battery. And finally, we have the pleasure to welcome Professor Dr. Tamara Tal, which is leading the mechanistic toxicology group at the Helmholtz Centre for Environmental Research. A group is developing NAMs for INT and DNT, and she's leading the PARC project aiming at developing further those NAMs. So we will listen to those three presentations. I will break briefly after each presentation, maximum 5 minutes, to give you the opportunity to ask clarifying questions, but only clarifying questions. And after the third presentation of Tamara, I will open the floor for discussion. So the aim is to have at least 30 minutes for discussion. So please be brief when you have questions for clarification. And now I will stop sharing and give the floor to the first speaker, Dr. Fritsche.
So thank you very much for this kind introduction. And I would like to start with really thanking ECHA for hosting us for this science seminar for the neurotoxicity. So we really appreciate the dialogue with you guys and are looking forward to this collaboration. So we have divided this time in between the three of us, and I would start out with the introduction on the topic. So it's not moving. Okay. So neurotoxicity is defined by adverse effects of compounds on structure or function of the nervous system. And here we distinguish between developmental neurotoxicity, where exposure takes place during development, acute or in a chronic way with adverse outcomes during critical phases of development. And it's important that it damages the construction of the brain architecture. While adult neurotoxicity has exposure after construction completion, so after development is already finished. And we distinguish between acute and chronic neurotoxicity, central and peripheral neurotoxicity with impairment of existing structures. So like was already said, DNT and ANT are regulated in very different ways. While DNT assessment so far is trigger dependent, the adult neurotoxicity is covered by the specific target organ toxicity, single or repeated exposure, and is actually tonnage dependent. So completely different types of regulations. Now, when we have developmental exposure, in humans, then we might observe neurodevelopmental disorders as adverse outcomes. For example, autism spectrum disorders, attention deficit hyperactivity disorders, schizophrenia, IQ loss, depression, or psychosis. And all these neurodevelopmental disorders have been increased in their prevalence over the last decades, and chemicals are suspected to contribute to the increase in the prevalence. So the structural defects that we see in these neurodevelopmental diseases, and for example, could be microcephaly, white matter defects, just to give two examples. And the important thing is that the probability is very high that these changes are irreversible. And it's now, there is a common understanding that these defects are based on interference with so-called key neurodevelopmental processes, which are depicted on the right side here. And to these belong the proliferation of neural precursor cells, migration, differentiation into neurons and glial cells, synaptogenesis, neurite growth, myelination, and in the end, neural network formation and function that forms then the circuits that are necessary for normal brain development. And due to the importance of these key neurodevelopmental processes, there has been the publication of a so-called generic DNT adverse outcome pathway, short AOP. And I would like to draw your attention to these cellular responses here in the middle of the AOP, which contain exactly the processes that I showed you on the slide before. And this is actually the basis of the current developmental neurotoxicity in vitro battery. So as biology as the starting point of these NAMs. So in contrast, the adult neurotoxicity, and I start with the acute, is actually acute exposure, like I said, on existing structures, and the existing structures will be different neuronal subtypes. We have astrocytes, oligodendrocytes, microglial cells, but we also have the different barriers like the blood-brain barrier and the choroid plexus in the brain. And the outcomes might be effects on sensory or motor functions, on the autonomous nervous system, but also on cognition or mood, like depression, for example. Whereas the chronic exposure is causing more long-term and very often low-dose exposure effects, and these include neuroinflammation, neurodegeneration, and especially the human diseases like Parkinson's and Alzheimer's disease, just to mention a couple, which are of concern, because in the end we would like to be protective for human health. And like I showed you now the cellular target, I would like to just briefly glimpse on the molecular targets. So we have all the different neurotransmitter receptors and transporters, and we have ion channels. So we have a lot of neuron-specific molecular targets that can be addressed by neurotoxic compounds. But also, in addition, we have cellular targets like SH group binding substances like methylmercury, for example, or interaction with cytoskeleton, mitochondria, ryanodine receptors, radical formation, myelin binding, and formation of neurofilament adducts, just to mention a few. So this is not supposed to be comprehensive. In addition, I would like to draw your attention to these hallmarks of neurodegenerative disease, because these have been identified as being a very important hallmarks for neurodegeneration. For example, defects on synaptic and neuronal networks, or altered energy homeostasis, which are mitochondrial toxins. And the interesting part here is, from the mechanistic point of view, that although all these hallmarks are important, they have different distributions in different neurodegenerative diseases. For example, here in Alzheimer's disease, you see that the mitochondria don't really play a crucial role, but they do in Parkinson's disease. And from these, and these are two examples, but there are more in the publication, this can be nicely used as a basis also for the biological applicability of NAMs. Now, I actually modified this generic DNT AOP to a generic ANT AOP, but I didn't put all the molecular information in here because the list is very, very long. But I think having such a generic ANT AOP more elaborated also during the phase of the new PART project will be detrimental for understanding applicability domains of ANT NAMs. So, and I would like to also draw your attention to a brain structure which is maybe not that prominent in neurotoxicity knowledge, and this is the neurogliogenic niche in the adult brain. And here we have the subventricular zones, but also for humans, this striatum, as areas in the brain which have adult neurogenesis. So that means up to the age of 80 or even beyond, neurogliogenesis in humans take place. And also neuron migration is depicted here on the right, and there's the age of the humans on the bottom, takes place until we're 80 years old. And there is a species differences in the in adult neurogenesis between mouse and human, which has been identified with the carbon-14 method. And it's really interesting that in the dentate gyrus of the hippocampus, so this is the place in the brain where learning and memory takes place, only 10% of the neurons are exchanged during the lifespan, or this is the first 12 months of age in the mouse. While in humans, this is actually very interesting. By the age that we are around 50, and I guess a lot of us have reached this age already here, 100% of our hippocampal neurons have been exchanged. So the human hippocampus is very plastic. So there's a lot of learning going on even until a relatively older age. So, and I would like to now get to two examples using the information that I showed you before. Example number one would be ethanol. So we all know that ethanol is an adult neurotoxicant. So how does it work? Through binding and activating to GABA receptors, but also inhibiting glutamate receptors. Then it also exerts oxidative and ER stress, and it inhibits adult neurogenesis. So when we drink just occasionally, we hope that these effects are reversible, but we know that when there is heavy drinking, so chronic abuse of high doses, it's ethanol can be very destructive with persistent changes like the Wernicke-Korsakoff syndrome or other encephalopathies after chronic high dose consumption. So how does it look for developmental neurotoxicity? So the fetal alcohol spectrum disorders is a very well-known human disease, and the mode of actions are the same, acting through GABA and glutamate receptors. However, even at low dose, so pregnant women shouldn't even drink 1 glass of alcohol. Persistent changes can already happen causing actually effects in synaptic plasticity and result in many psychiatric disorders. So that means that the exposure during adult and developmental is really in their effects and their reversibility very different. So the second example is retinoic acid because it causes neural tube closure defects, and we know that that in the adult, this mechanism is not relevant because the neural tube, in the best case, is in most cases already closed. However, we also showed in our recent publication, and this was an EU project, that retinoic acid also impairs neuroprogenitor cell proliferation through the retinoic acid receptor, which is shown here. And this was actually the most sensitive endpoint across a variety of DNT in vitro battery assays. So what about adult excess retinoids? So they are linked to depression, mood disturbances, and cognitive defects in some patients. And in rodents, it was shown that retinoic acid also suppresses adult neurogenesis, and this is BRDU incorporation, which is actually the same measure than we do in our proliferative progenitor cells in vitro, and thereby also triggers depressive behavior. So that means here to distinguish between the same mode of action and the effects on the developing and the adult brain is not really that easy. So the hypothesis would be that we came up with in this new PARC project is that ANT mode of actions can also trigger DNT usually, because all these targets here are important during development. But the question is if DNT mode of actions might, or with reduced potency, trigger ANT. So, and how large is the common space of DNT and ANT, and how can we distinguish qualitative and quantitative ANT commonalities? So this is really something which is unknown that we would like to explore. And so final thoughts on putting this project together from my side were that timing might be the main determinant of DNT versus ANT, so developmental versus adult exposure. But ANT effects are also more likely to be reversible, like with our, from time to time glass of wine consumption. But for sure is that the developing brain is more susceptible towards insults than the adult brain. And the question is a qualitative distinction between DNT and ANT even possible? But these are all questions that we would like to address in this project, and of course with the central question if NAMs can delineate between DNT and ANT. And while I gave you a glimpse into phase 1 of the project, which deals with hypothesis generation, AOP, landscaping, and also especially looking for mode of action, this will be in more depth being widened in the first phase. And with this, I'm coming to the end, and I'm looking forward to the discussions. Thank you very much.
Very interesting introduction and a lot of interesting questions. I think there is one question online. Yes, Kai, please. So now I'm opening the floor just for clarifying questions. So we have a bit of time. Thank you, Hélène. You made it on time. But please, Kai, go ahead.
Thank you, Ellen, for the very nice introduction. If we're going to have a discussion on adult versus developmental effects, my clarifying question would be, what is the consensus in the PARC community on when brain development ends? Because you need to be able to draw the line somewhere. And as you discussed, the subventricular niche, like there are processes still ongoing. So where do you draw the line? When does brain development end? Thanks.
So, I mean, this is, thanks a lot, Kai, for this clarifying question. Actually, we discussed this yesterday at the PARC stakeholder meeting, exactly this question. So scientifically, brain development ends when the juvenile or a young adult is turning 25. So this is a scientific consensus, because still brain development during puberty, for example, and then later on until we got full maturity, and are not very likely to to rush with a car, you know, in dangerous situations really takes some time. But I think this puberty phase, I'm not sure if this is currently covered very well in the guidelines, because this is far beyond, I think, just a learning and memory example. And we are very well aware in the scientific community that also this teenage phase, for example, is not really covered well. And there are initiatives out there right now to try and acquire also additional EU funds for actually setting up NAMs for this phase, because this is very specific. And I'm not even sure if this crazy teenage phase is well reflected in rodents, but maybe you know more about this.
So I propose we move on, and I think the next speaker is Jonathan Blum. So Jonathan, the floor is yours.
Yeah, I will dive directly into where Ellen stopped, which is a new proposal for the final 3 years of PARC, where we currently work on. As Ellen really nicely highlighted, I mean, it's a hypothesis, and this hypothesis needs to be tested, and we want to be really careful to do this in a strategic way. This is what you've already seen. So we really want to, before we dive into a testing phase, want to make sure we have a good hypothesis, a good idea how this could look like. And part of this is doing desk research and searching for what is out there. And partially, this is also data of NAMs, which is already produced. And this will then hopefully guide a real clear phase of testing, which will be linked to a previous project in PARC. And you will hear from Tamara about that a bit more. And yeah, I think what's really great about this new project idea is that it's such a close collaboration with regulatory bodies, mainly ECHA. So how we set up this phases is also really closely linked to something published this year by ECHA, which is key areas of regulatory challenge, where neurotoxicity takes a big part in as needing further research and needing research on NAMs and how NAMs can deal with this and hopefully solve some issues. And so there's a very clear link between this document and how we designed the project. And one aspect of this needs defined in this document is work on AOPs, and Ellen already mentioned it. And the AOP world can be complex, and we really want to dive as deep into it as possible, and with the full complexity. So there might be networks, common key events, and shared or different adverse outcomes between different AOPs. But we really want to link then this regulatory link from adverse outcomes to key events which are testable. And this was already highlighted nicely by Ellen. So we know what we can do with the NAMs. We have the battery, we have an idea about ANT NAMs, and we really want to feed this information into existing networks on neurotox AOPs, which are already published, but we plan also to expand them, of course, and add information to these networks, which will help us to address this scientific question. So one example, what I mean with adding information, is as simple as highlighting developmental stage. So the timing, which already mentioned, I'm really happy that this is also reflected in the document by ECHA. Timing is a crucial role for DNT and for ANT, and it's not that much reflected in AOPs classically. So we really want to bring this in. So if the events are considered old age, early age, developmental. So this is one aspect, and this is work currently performed by PhD students in our institute. And of course, a lot will build around very established AOPs. So I picked AOP3 as an example. It's one of the most established ones, OECD endorsed, and it's linked to Parkinson. And stressors are known to cause this. So we would consider this an ANT. And then you can build a network around this AOP. But yeah, we listed here, and then this is now an example case where we really have to do research then on these AOPs. So some are basically empty entries in the AOP wiki database. So this is what we mean with proper desk research, to really put into a network what we consider useful to address this question. And I also want to use this AOP then to give an example how we consider how it can help us then in a test phase to address this question. So we have cell types with partners within this project proposal where we could address this event of dopaminergic neurodegeneration. So this would be a classical ANT consideration here linked to this AOP, but we actually can treat a different time frame of cell line development, and this is an assay part of the DNT battery. So this would be an example how we think these AOPs can really help us guide the testing, come up with an hypothesis, and then potentially test this overlap, which Ellen depicted in the end of her talk, that a lot of ANT potentially will overlap with DNT. And another example, this is then again the timing, that same molecular events in different cell types may lead to different adverse outcomes. And this is also something where we think we could test this, but we need to feed this information into the AOPs. So neural crest cells are during this phase of neural tube development, which Ellen mentioned. So a classical assay in the core battery, but it triggers the same molecular initiating event, which may also feed into this established ANT AOP. So these are just examples of how we plan to integrate NUM testing with AOPs and then linkage to adverse outcomes, and hopefully understand better the overlap and the differences here. So the overall picture might look quite complex then in the end, but this is really what we will use in this project to understand. And yeah, just picked some examples here I mentioned already by Ellen, that the developmental stage and the cell types and so on will play a crucial role for understanding the processes here. Then another need is reference compounds, and this is also something where we know how difficult it can be from the work with the developmental neurotoxicity in vitro battery, where we chose positive and negative control compounds to validate the battery based on literature available. But there's more, and there's updates, and we really want to align and compare and make this list as extensive as possible. And then, of course, to address this question, combine it with ANT lists. And here, ECHA already helped us out with a recent publication on a reference list for neurotoxicants based on CRP classifications. So this is really helpful, and a lot is already in there, and also to comparison to DNT IVB compounds. So we will definitely use this, or plan to use this as a starting point for our investigations. And on this, also started some work. A master student that is working on this. Of course, this list is based a lot on the harmonized classification, but coming from the AOPs, and what we want to test with our NUMs is a lot biology based. So I picked as one example the AOP3 and the compound rotenone, which I've shown, and which is not in this list. But of course, it would be a good two compound. So we have to see how much we will use this list, and to which extent yeah, we want to extend it. So chlorpyrifos is another case. So Tamara will show something on it. So organophosphate compounds are in this list, but then it depends, as Ellen said, also on the tonnage and on the classification situation for some compounds. Yeah, so we might extend this list. So we currently look into this. And yeah, then we want to use what we have from the battery. So of course we will do a lot of database search. So this will be a big part of the project. And I picked now 2 examples, which I thought give a glimpse of what we might think, which direction it then could go in specific case studies. And Tamara will show more about this. Which is linked to what Alan has shown with retinoic acid. So this is really early phase. So this is an ECHA publication on human brain development. It's actually not a tox paper, but it's really reflecting what we try on a mechanistic way to do with the battery. And there's this early process of neural tube formation. And Alan showed examples, and I will show 2 more examples how this could be addressed. So we have partners in the project where we could even run rosette formation assays that reflect this process. And this is not part of the core battery because not one of the higher throughput assays are not as far in fit for purpose validation. But it could be used for such targeted case studies, which we plan in this project. And retinoic acid, we know, is hitting the cells in a way that they differentiate into endothelial cell actually, and are not able to form these rosettes anymore. And then there's neural crest migration, which is also within this time frame. The retinoic acid is also hit. So as an example, how we consider to really guide this mechanistic testing, and this could be a potential DNT only mechanism, but as Alan explained, it can be difficult, and retinoic acid can also cause ANT to some extent. And another example at the late stage I want to show is then a microelectrode array assay was established with partners in the project. And you will see more from Tamara on this, but it's a great example how the treatment time may help us answer both questions. So this is a publication for a DNT setup of a microelectrode array where you can test for network formation, but of course you can also have advanced networks and assess toxicity of these networks. So this hopefully gave a glimpse of what we want to do. And we will build a lot of the test phase based on the previous project. And it's great that we have the project lead here. So I will hand over to Tamara here. And this is a quote I stumbled when I prepared the slides. It's not clear if it was Galileo, but I think it really fits in the direction we want to go. So we really want to measure and investigate what we can measure with NUMS, and where we see gaps and needs, we will try to make it measurable. Yeah, so with that, thank you, and also look forward to the questions.
Thank you very much, Jonathan. Very interesting to see that our CARC is used by PARC and by the scientific community to develop projects. So really we are happy to see that it's useful for you, and it will be useful for us as well. Is there any clarifying question? For now, I'm just taking question to clarify the presentation.
I see one in the chat.
There is indeed one in the chat. I don't know if Elizabeth, would you like to maybe phrase it?
I can try to answer it. So, as I mentioned, I was really happy when I read this also in the ECHA document, because how AOPs are designed, the time is often a bit neglected, and this makes sense for a lot of questions to address. I mean, a liver cell sometimes, but for our question, it is highly of high importance, the timing, and that's why we need to implement this somehow in the AOPs, and that's why we want to refine this networks. So it could be that there are early oligodendrocytes and they behave different than developed oligodendrocytes, and this is often not depicted in AOP. So we plan to add this information, and the same applies to different cell types. And yeah, I try to give 1 or 2 examples on how we want to address it with the in vitro assays. So we can test early oligodendrocyte differentiation. We can test late stages of oligodendrocytes. We can test developing dopaminergic neurites. We can test on an advanced network. But I think this is really where then the hypothesis part comes in. We have to investigate this, and this is where we really look forward to do so. I hope this answers the question.
Yes, thank you very
Maybe I
much.
can add to this that especially the clearer definition of timing and cell types and processes of the key events is also part of the OECD AOP work. So the special knowledge interest groups and stuff. So together with the JRC. So I think so this will be also addressed in an international context in parallel to PARC. So and there are connections between.
I suggest we move on to the last presentation from Dr. Tamara Tal. Please, the floor is yours.
Thanks very much for the opportunity to present today and to start this really what I think will be a fruitful exchange. I'm taking the angle of focusing on how, extending off of what Johnny just presented, to discuss how we might approach answering the question using specifically NAMs that have been developed within the PARC project 5.2.1e. So first, in the DNT context, it's really important to understand when we're building new NAMs what we're aiming for. And the gold standard for NAM-based DNT testing at the moment is the DNT in vitro battery that has been mentioned in both of the previous talks. This is comprised of 17 assays that are mostly human-based, although there are some rodent-based assays that are performed in cortical preparations from pups. And they focus, as Ellen mentioned, on key processes that facilitate neurodevelopment. And recently, there is an OECD document 377 that defines the initial recommendations for use of the DNT IVB. The current regulatory status of the DNT IVB is that it's under public consultation within the plant protection product legislation, where in an annex it states that DNT shall always be assessed, that in vivo guideline testing can be waived if a substance is negative within the test battery, and a weight of evidence assessment shows reliability and there are no other concerns for DNT. If the substance is active in the DNT IVB, then this annex recommends quantitative exposure considerations need to be carried out, as well as additional qualitative considerations that can be included as part of the overall weight of evidence. And it says at the end that additional in vivo DNT studies shall only be undertaken when it is not possible to carry out a risk assessment based on all available information. Of course, this is still under public consultation, but this shows how there is a line to implementation for the DNT IVB underway. But really importantly, within that OECD 377 document that was reviewed by ECHA and many other regulatory agencies, it was really clear that there are uncertainties and that the battery needs to evolve as the science evolves. And that really set the stage for the PARC 5.2.1e project that was designed to look at a potential expansion of the DNT IVB and to assemble first generation adult neurotoxicity in vitro battery. And in the context of the DNT, the idea here was to improve existing NAMs to reduce costs and increase throughput. Some of these tests can be lengthy and costly. To build new NAMs that cover uncertainties, for example, key neurodevelopmental processes that are not reflected within the current DNT IVB, for example, assays that cover myelination or blood-brain barrier development and function. To build confidence in the use of the DNT IVB, that these NAMs cover key modes of action in a DNT context, for example, endocrine disruption, and to increase the readiness of these NAMs by evaluating them against a common library. It is not our intention to create an ever larger set of NAMs that comprise the DNT IVB. Instead, it's to identify the minimum set necessary to adequately predict DNT. And the way that we as a team have come up with our approach is to generate NAMs with high readiness, but to evaluate their ability to add value through testing a common library that was generated collectively and then procured and provided by the JRC, and testing is underway on a 96-member chemical test set. So I'm going to share just 2 quick case studies using NAMs that have been generated within the PARC project to show you how you can get kind of consistent or different data. And the first case study focuses on chlorpyrifos, which is an organophosphate pesticide. And as Johnny mentioned earlier, this is a mode of action that is of interest from an ANT context, although this particular chemical was not listed within the Crane and ANT reference list. So within that same paper, it says substances that are registered under the biocidal products regulation or the plant protection product regulation can be assigned to 6 major categories where the neurotoxic mode of action is known or assumed based on their mechanism. And this includes the organophosphate pesticides, again, which chlorpyrifos is one of. So when this compound has been evaluated within the DNT IVB, it was negative, and also the transformation product chlorpyrifos oxone was negative in the Blum version of the DNT IVB. So this is something that's a little confusing maybe in the literature, is that there's 17 assays, and some papers use some of the assays and not all, because these were, as you all probably know, many of the assays were generated in 3 main academic and government research labs. This compound was positive in the US EPA microelectroarray assay from Tim Schaefer's lab. So we've tested it now in a new NAM that was developed in my group through the PARC project. This is the zebrafish visual and acoustic motor response NAM, the VAMR NAM. What you're seeing here is the behavioral profile of all of our historical controls. This is over 5,000 individuals. And on the y-axis is the motor activity in pixels per second. On the x-axis are 26 endpoints that indicate different stimuli, either visual or acoustic stimuli. And the idea here, what's popped up, is this classic light-dark transition. So the fish freeze when the lights come on under this visual motor response 1 assay, and when the lights turn off, they increase their activity, and then it slowly decreases over time. I'm just going to highlight a few key endpoints I think are interesting to discuss. We also, because we're returning the behavior data on a per second basis, capture startle responses, and this is a behavior that is conserved in zebrafish up the evolutionary scale to humans, and we have startle responses in response to visual stimuli, so dark to light or light to dark transitions, and also acoustic stimuli. And the last endpoint I'll highlight is a learning related endpoint, which we just heard a lot about in both of the previous talks. So this is reapplying the same acoustic stimuli 30 times with a very short 1 second interstimulus interval. And the zebrafish learns not to waste energy responding to this non-threatening stimulus. And this is called non-associative habituation learning. There's 10 key criteria you have to demonstrate to show that this is learning and not just fatigue, which we've done in one of our first papers establishing this assay. And you can see the smooth spline for this response on the right-hand side of the slide. So these are endpoints which complement the process-based endpoints in the DNT IVB by adding functional endpoints and kind of span endpoints that have potentially more orthologous relationships to the test guideline studies that we ultimately seek to potentially replace collectively as a battery. So back to chlorpyrifos, we looked at this compound under 2 exposure modes, and this really gets back to what Ellen and Johnny have both been talking about, that we need to consider timing when we start to look at this question of whether NAMs can delineate between DNT and ANT. So this is under an acute mode where the compound's applied for 40 minutes before testing. And you can see on the bottom of the slide, the control group in gray and the chlorpyrifos exposed group in green. And what you can appreciate is that a short-term application of this chemical causes widespread differences in behavior across the entire endpoint battery within this one NAM. Zebrafish offer rapid testing with medium to high throughput, so we're able to test many concentrations. Just for one endpoint shown in figure A, this is the interstimulus interval. This is a normally quiescent period, and here increasing concentrations of the compound provoke inappropriate locomotor activity in this normally quiescent period. And then on the right, you can see the concentration response profile following the acute application of chlorpyrifos across the 26 endpoint assay. And you can see these really interesting patterns that begin to emerge. And these patterns, when put up against a huge battery that we have of reference compounds for known critical DNT modes of action, provide a fingerprinting approach where we can quickly identify neuroactive chemicals and predict their underlying mode of action that can then be empirically evaluated. But whether changes in neuroactivity really reflect developmental neurotoxicity is an open question. So through the PARC project within the screen, all of those compounds are being screened under a developmental neurotoxicity exposure paradigm. Here, the compounds applied at 1 day post-fertilization. So neurogenesis has begun, but it's not completed the primary phase where the nervous system is kind of mapped out in a body plan until 3 days post-fertilization. The chemicals present from day 1 to day 4. We then wash the chemical out and 24 hours later perform the behavior test. And here what you notice is two things maybe. One is that as opposed to having a pattern of activity profiles where you have some endpoints that are down, many that were up, here we see consistently reduced locomotor activity across many, but not all of these endpoints, shown as an example on the bottom for the visual motor response 2. This is the dark phase. Another thing you might notice is that this testing under DNT mode enhanced potency detection. So it was about 2 orders of magnitude lower to see significant effects following a developmental exposure. And that phenomenon where you see enhanced potency detection with the developmental exposure paradigm happens a lot. But I just want to show you that it doesn't always happen. So this is another compound, ketamine. This is a reference chemical for NMDA receptor antagonism, and this is a classic way that you can impair habituation learning in rodents and in zebrafish. And on the top, what you see, and maybe I use my pointer finally, under the 40-minute acute exposure paradigm, what you see is what you would maybe predict based on the rodent literature, the controls in gray, and you prevent this habituation learning response following exposure to ketamine. And really interestingly, under the developmental exposure paradigm, so again, compound on at day 1 and off at day 4, 24-hour washout, and then testing, this compound is also causes a significant effect or at the level of habituation learning, but instead of impairing habituation learning, you have inappropriate habituation learning relative to the control. And what you notice on the right is that the concentration at which you see these changes isn't very different. So I want to make the point that the acute versus the developmental exposure paradigm is often different, but not always. But what you gain from the acute exposure paradigm is that you can get more of an understanding into what the potential underlying mode of action might be. So for the second case study, this focuses on PFAS, this is a per and polyfluoroalkyl substance. It's been restricted in the EU since 2006 and included in the Stockholm Convention in 2009. And I'm going to start now at the level of the MEA assay, but I actually don't want to present Ellen's work for her. So I'm going to just quickly invite Ellen to present the next 2 slides.
Thank you very much, Tamara. So I'm jumping in because this is coming out of my old lab in Germany. And so this is actually the brain sphere system, and these are human-induced pluripotent stem cells, which we perform neural induction and put them onto a shaker and produce then 3D brain spheres that differentiate during the shaking mechanism, and that we can culture in 96 well plates and also place on MEAs. So the MEAs that Tamara introduced beforehand as a functional readout for neurotoxicity. And maybe you appreciate this little image down here. This is a spike raster plot where on the x-axis you would have the time, and on the y-axis these are the different electrodes, and the blue ones are areas of high activity, so that means that the cells are bursting and they are fairly mature. And when they are synchronized, then you know that you have a network that is matured. So now you can imagine that many neurons, they grow over one electrode, because like in a 96 well plate, you have like 9 electrodes. It's not that many. So that means when you display a signal, and this is the gray on the very right up here, that you might have signals that come from different neuronal units, and you might not be able to depict a certain effect of a compound because there are different neuronal subtypes. And but we have something that's beautiful. It's called spike sorting, and this is the colorful picture that you see on the bottom, and that means that this is actually a software which you do after the recording, can distinguish between different spiking shapes. And so by analyzing individual spikes, we can discriminate between different neuronal subtypes and so understand the mode of action in the system. I think we can put the next slide. Oh, and it can, and thereby we have now been able to show that we have GABAergic, glutamatergic, dopa, choline, and serotonergic neurons in the system, in networks, in co-culture with neurons and astrocytes. And to understand the development of these neuronal subtypes in the system, I'm not going here briefly into the detail, but on the very left, you have a gene expression profile concerning GABAergic, next to it, the glutamatergic, then the dopaminergic, and on the very right-hand side, cholinergic, and also here's just one gene for the serotonergic. And what I want you to appreciate is the red coloring, which for the GABAergic on the very left, and this is actually gene expression over time from left to right, starts developing kind of in the middle of the column, while for the dopaminergic, this is the third from the left, you see that the red is more centered to the right. So we've been monitoring here over a 28-day period. And that's important to understand when the neurotransmitter system actually occur in the systems to understand when you run your functional test then for ANT. And we have been actually setting up this ANT assay within the PARC project. And so Katharina Koch is continuing this at the IUF. And I give the word back to Tamara.
Thanks, Helen. So as part of the PARC collaboration, we've been working on PFAS chemicals for a while, and we found a mechanism in zebrafish, but I want to start the story actually in the human MEA neurospheres. So Ilka Sharkin was part of that group at IUF, looked at PFAS exposure within the brain spheres on MEAs and showed that exposure to between 100 to 120 micromolar PFAS in this case basically increased the normalized spike count. So you had a more active neural network. And this is consistent with the hypothesis that this compound antagonizes GABA receptors, which are the main inhibitory neurotransmitter system in the brain. So this, again, you would predict then you would see an increase in activity. And that's what you see here. We took a look at this compound in the VAMR assay in the fish, and I'm showing the visual motor response too. This is the dark period, which is historically very sensitive to PFAS exposure. And we see, again, what you would predict under a developmental exposure paradigm, so the chemicals on at day 1, off at day 4, testing at day 5, that you have elevated motor activity, again, consistent with the antagonism of the GABA A receptors. And on the right, we've spent a really long time figuring out how to fit these heterogeneous behavior data so we can put them into the US EPA TCPL2 fit pipeline to generate point of departure estimates or AC50 values so we can compare the potency of these compounds across the different assays. And then lastly, we want to use the NAMs, but we also want to build as much confidence as we can around the findings that we have. So we often will team up with our colleagues for orthogonal evaluation in different systems. So this is data from my colleague Anna Nerlich at University Leipzig. And she took cortical mouse neurons, electrophysiology experiments, and showed really clearly that here's the GABA-related current, and it's completely blocked when PFAS is present in the system. So we have it in 3 experimental systems under varying exposure regimes, human, mouse, and zebrafish, that this compound acts as a GABA receptor antagonist, and that this mechanism seems to be conserved. So the final strategy, and we really look forward to this iterative collaboration with ECHA as we formulate the questions, identify the case studies, and test them, is, again, can NAMs delineate between DNT and ANT? And there's some text that seems to, it's not coming up right at the beginning, sorry. So following the identification of testing gaps through the literature surveys and also scraping and using all of the existing data from sources like the US EPA CompTox and Tox21 databases. We want to identify testing gaps, and both from an assay perspective and chemical perspective, and then we want to use PARC 5.2.1e DNT and ANT NAMs to perform gap filling case studies, where we, for example, test the crane and ANT reference chemicals in DNT assays, and we evaluate DNT reference chemicals in ANT assays that have been generated within our PARC project. And just one example here that's already been alluded to, this is the MEA assay, and you've seen very short acute exposures in that assay up until now, but here's how this assay, and it's under development at IUF in the Koch lab, to basically create a mature network, do a kind of chronic exposure model with a 7-day exposure, and then perform this network analysis. So with that, I'll end. Thanks very much again for the chance to present, and I'm looking forward to your questions.
Thank you very much, Tamara. This is impressive what you have achieved on the PARC. So now looking at the time, I think I will already open the floor for discussion, but of course the audience can also ask questions for clarification on your presentation. So now the floor is open for any questions.
We are also happy to receive input on the project. Suggestions are welcome as well.
Okay, thank you, Kai. You can go ahead, please.
Yes, thanks. Yeah, very interesting presentations and interesting upcoming work as well. Feeding back to the original hypothesis that was raised by Ellen. So if I understand correctly, the hypothesis is that essentially all ANT substances will cause DNT, but not necessarily vice versa. And then the question is, how big do you set the domain of which types of substances fall under the ANT norm, correct? So my question is that then in theory, every ANT substance that is run through the DNT IVB should be picked up as DNT as well in one or more of the assays. So what if you test an ANT substance and it's not picked up as a DNT substance? So how will you address that in your discussion at the end of this project? Because There could be any number of reasons why it's not picked up. It could be that it's outside of the applicability domain. It could be that the assay simply failed to deliver. It could also be that the hypothesis may need to be revisited, that there are ANT substances that are not DNT. So how do you intend to address this? Because this question may pop up multiple times. Thanks.
So thank you, Kai. I think what you just outlined is a beautiful outline of our hypothesis and how we're going to address this, because at the end of the day, we formulated this as an hypothesis because we want to test if it proves correct or if it proves wrong. So it might very well be that an adult neurotoxicant is not a DNT compound, but the only way to understand this is by understanding the mode of actions, setting up the respective AOPs, looking at all the literature available. If there is not the right literature and not the right data to answer the question, we have to generate, but this is really the last resort, we have to generate new data. So this data, it depends on what it is. I mean, it could range from, is the potential target even expressed in the system? I mean, we have so many RNA-seq data by now that this is something we should be able to address relatively easily in the NUMs that are currently available, not by producing new data, but maybe even a target changes upon exposure, induction, repression. So I think we have to kind of what EFSA is calling an IATA approach. I think we have to do, and this is basically our most interesting case studies then, but I mean, you outlined the three possibilities. I mean, outside of the applicability domain, I think, and maybe hypothesis is wrong. I mean, these are the two most prominent possibilities for me.
Thanks. I mean, I just very quickly want to catch up to that. So looking back at the applicability domain, when you look at this list of ANT reference substances that we recently published, so there's 113 of them. And a part of those, the mode of action, as was already stated, is either known or can be assumed based on what the substance was designed to do. Like many of these are active substances in biocides or pesticides. But many other substances, there's a very poor understanding on the mode of action. So it might be more difficult to decide which assays you want to run them in. And then there's also the fact that many of those substances with a poorly understood mode of action, or with a very diverse mode of action, it doesn't need to be specific per se, can be organic solvents. They can be very volatile substances. It can be very difficult to test them. So I think at the very onset of this project, it would be good to have very clear exclusion criteria why some things would not be tested. And these would then be classes that essentially at this stage fall outside of the hypothesis because you simply cannot test them. Thank you.
I mean, I would say these fall out of the chemical applicability domain. And at the UFZ, Tamara, you have the expertise, right? So.
Okay, well, this was done in collaboration, of course, with you and Stefan Schultz, and the 96 chemicals test set that ECHA has procured for us underwent a very strict qualitative criteria to select these test chemicals for exactly those kinds of physical chemical properties that would prevent testing within our test systems. But I did want to point out that if you reduce the throughput, you can test semi-volatile compounds in the zebrafish assay when you use a hermetically sealed exposure vial, and that's been recently published for quite a few pretty volatile pH-related compounds.
And I would like to add towards your applicability domain and the mode of actions. So in 2018, we published a common work with EFSA, that was an EFSA procurement on the question of adult neurotoxicity. And EFSA proposed, so these were more than 200 compounds, including natural neurotoxins, And we did a mode of action analysis with the current databases available. And these cover a lot. So I think the first thing I would like to do is also compare the compound list with this EFSA project with the ECHA list, because we have in a table grouped if there is one or if there are more mode of actions, which ones they are. And from the mode of actions, we assembled the applicability domain that an ANT NAM should have. So this is data that happened almost 10 years ago, but we still will be using this.
Okay, thank you very much. I see two other questions. First is Camelia, please.
Thank you very much for very interesting presentations, really very nice work. But I was just wondering, because we as regulator, we need to identify the hazard first for the risk assessment and then to use this data for classification purposes, because in the end, that's the final goal, right? How do you think that these batteries can be used for classification purposes? If I recall correctly, you had on some slide that if the results are negative, then that's it, we stop following the hazard. If we have some positive results, then what's happening then? Do you envision in vivo testing, or what's the plan?
I think you refer to the annex of the new plant protection product legislation. So what's actually the EU Commission is proposing there, but we don't know if this is the final version, is when we have a positive, we have to collect all the available data and perform a weight of evidence assessment. And it's also written that only as the very last resort, when a risk assessment decision cannot be taken, then animal experiments might be necessary. But they're not writing that it should be the full like OECD 427, but I'm thinking probably more towards also a targeted assessment might be necessary. But as Tamara said, so this is not the final version of the legislation because it's open up for commenting right now. But I think the most important thing is when you deal with these DNT or ANT ants is that you are very certain not only about the chemical applicability domain, but the biological. And this is why we're really stressing to understand the different modes of action. For the adult neurotoxicity, this is much easier than for DNT, because for DNT, at least when we started working with DNT, all the MIEs or the target structures were not really known or comprehensively described, whereas ANT, you have much more textbook knowledge. And so there will be overlap, yes, and because all these targets that I showed for adult neurotox, they're important for early circuit formation, for early brain development, like you saw with the ethanol, for example. If you mess up GABA and glutamate signaling, you can have detrimental results that are not reversible because they mess up your early circuits. So I think understanding that your NUMs cover the mode of actions that are possible, I think that is the most critical to decrease the uncertainty.
That's absolutely correct. I was just thinking more from a practical perspective of reach. I'm not referring to another legislation, just to reach. I was wondering, I mean, if we think about the sensitization IATA, for example, you can classify on the basis of the in vitro data. So I think the same should be applicable when this system that you are working now on is going to be validated and it will be certain enough to be used. So just wanted to make sure that actually in the end of the day, it will be possible to take a decision, a clear decision that it should be useful for classification purposes, because at least in the context of REACH legislation and chemical legislation, this is a very important outcome.
So the outcome would be that there is a checklist for ECHA. And if you check the boxes, you know, if there is a hazard, and even if it's in vitro hazard, and then it's a hazard. So it should be used for classification, just like an in vivo hazard. I don't see that there is any difference, because we do have quite some compounds who do not mess with any of the endpoints. I mean, that does, of course, that does exist. I mean, even like a pesticide like flufenicid did not show up in the battery. It was an absolute negative, although it's an endocrine disruptor and it showed up in that assay where it should show up. So I'm pretty confident also with the negatives that we can use these NAMs if we understand the mode of action for classification purposes.
Yeah, thank you very much.
And it's why we really want to run this case study. So last week we discussed in Athens that the defined approaches as used for the skin, they kind of evolved out of the IATAs. And if we could go this direction, we would be really happy. Yeah.
Thank you. So now I'll move on to Cécile Michel.
Hi. Hello, everybody. Thanks a lot to ECHA for organizing this seminar. Thanks to the presenters for the very interesting presentations. I had a more global question that relates also to what Camelia just asked, and also some of the remarks made by Kay previously. I wonder, I am not as specialist as the three speakers, so I thought that Hélène might be a bit less definitive than I would be, but from my, as a toxicologist, it makes sense for me that any ANT would probably be DNT toxicant as well, and that you can react on that first statement. So if this first statement is true, for me, then the question is really more about how the system that you are developing will be sensitive enough to maybe identify chemicals that would be DNT without being ANT. But I don't know if we are here so far, especially because the chemicals that you presented in the presentation published by ECHA, for example, are about ANTs, and I was wondering if we know if it already exists a chemical that we know is not an adult neurotoxicant, but is a developmental neurotoxicant. And for me, it's probably also around the dose where those effects would occur, and it's then more related to risk assessment. And then the question is also about how in vitro tests can be used to identify those doses where DNT would occur without ANT. So I think it's the question is a bit different. So I think there is many questions in my intervention, but I was wondering to summarize it is why is it so important to distinguish the adult neurotoxicant than the DNT? Also, I appreciate that the CLP consequences are completely different, but in a scientific point of view, and also, are we mature enough to really distinguish? And I think that already having an in vitro model enabling to identify neurotoxicants, whether they are adult or developmental ones, would be already great. So yeah, thank you for your feedback.
I'm not really sure what exactly I should answer now. There were many, many different questions. And one, you asked for examples where the adult is less sensitive than the developmental organism. And that is clearly in the metal field, for example, that against lead or methyl mercury, I mean, where we have the human exposure due to the unfortunate incidences of exposure through food, we know that the developmental organism is much more sensitive. And maybe Tamara, with the zebrafish assay, has also examples with the sensitivity that she shows. So maybe Tamara, I give the word to you.
Yeah, I think with the assays that we have, we have a DNT assay that has the exposure that ends before testing, and then we have this acute assay. And whether this acute assay, which returns changes in neuroactivity that are likely receptor mediated because it occurs so quickly after exposure, whether or not this is a true ANT assay is really out for debate still. So we have not yet had many examples where we see complete activity in one mode and nothing happening in the other. But what we do often see is that there can be a shift in the potency detection.
I have one, so I read the question in the chat.
Yeah.
I have one example actually from our lab. So this is not it is a chemical, but it's also a natural chemical. It's epigallocatechin galate. So it's a green tea catechin that is added also to many different foods. And we know that it's not an adult neurotoxicant, but when given during development, we have identified the mechanism that it interferes with migration of radioglial cells. And so yeah, that would be for an example when something interferes with the building of the brain, like especially like migration or differentiation processes, that these are not necessarily ANT compounds unless you have very high concentration and chronic exposure. Then you might also have effects on the adult hippocampus. But going down these lines, Johnny, do you have also an example?
Go ahead.
I have a comment, and I think I have an example. So the problem from the DNT side is that we really struggle to have data-rich DNT compounds. So there's this list of human evidence DNT compounds, and it's like 5 or 6. So the really classical ones with human data. And one example is valproic acid, which is an anti-epileptic drug, which is used because epileptic seizures are even more severe during pregnancy than the drug, but it's known that it causes, for example, neural tube issues, which was one of the examples which we used. And I hardly believe it's on Kai's list from the paper. I think it's a classical DNT one, but there really is a limited number of DNT compounds, and the in vivo guideline studies very rarely performed. So there's also not a lot of in vivo data. So I think that is part of the problem why there's not a huge list of DNT only, but we will explore this. And this is now one that I have in my mind. Maybe there are some others. And then it comes back to potency, like methylmercury and the metals I mentioned.
Yeah, but just to quickly put a number on it, I mean, in the publicly available guideline studies, we took a close look at this, and recently Kevin Crofton's done it also, and it's 144 unique chemicals that have been tested in the test guidelines for DNT and in the rodent studies. That's publicly available in the ToxCast ref in the ToxRef database. But what we would need to test this idea is we would need negatives, so ANT negatives that we could then test within the DNT NAMs, and that would be a really great case study and really not terribly difficult to do because these assays are very well established and they're running smoothly in multiple groups.
Yeah, thank you. I think it clarifies also, I think the two examples that you provided are really about, a question of potency, as mentioned by Ellen. The name of the green tea active substance. I agree that at high doses, we know that it's going to affect brain, and for anti-epileptic drugs, for sure, these are their pharmacological reasons to be. So I think they are active neuroactive substance, not to say neurotoxic substances. So for me, it's really, it's a question about philosophy, about why to spend so much effort in distinguishing this question. Also, again, I appreciate that the consequences in terms of the regulatory consequences under the CLP are much different. But if we all agree around the table that adult neurotox should be per se considered developmental neurotox, it would save us a lot of money, time, and resources, and then helps you to focus in developing an in vitro battery that capture DNT and ANT, and I mean, who cares? Thank you.
So this is a great comment. So, I mean, that goes into the direction that maybe we do not need two batteries, a DNT and an ANT, but that we need a DNT battery that it's expanded for the ANT mode of action. Then we have one neurotox battery, because having only an ANT battery, I'm very afraid that we miss out on developmental effects, because I mean, that's the legislation what we have right now. And but having a DNT plus battery might be a solution. So, I mean, thank you very much for this comment. And I think we also take this into consideration, you know, when we, at the end of the project, you know, come to recommendations.
Okay, thank you very much. So, Kai, you are the next on the list. Please go ahead.
Yes, so actually, I think what Cecile said does resonate with me as a scientist. So I do think quite a lot of neurotoxicologists do consider that adult neurotoxicants likely also elicit developmental neurotoxicity. But I mean, open any kind of textbook on neurotoxicology, and what you will read at some point is that the developing nervous system is more sensitive than the adult nervous system. But the classification paradigm that we have right now does force to make the differentiation. And if you want to open the discussion on whether the current classification paradigm is appropriate for neurotoxicants, then you need to have a data-driven approach to open this discussion. And I think that's why we are all here today, to look into this, because it's easy to make assumptions, but I don't think anybody has actually ever in a systematic way tried to address this hypothesis. It's something that you're taught in uni, but I don't think anybody ever actually properly looked into this. Thanks.
I can give the floor to Niklas.
Thank you for a very interesting presentation on a very challenging topic, and I think there's a lot more to be done on all angles of this to expand our knowledge about developmental neurotoxicity in general. And I think my question is just a bit more practical. So I get this battery as a regulator. What do I do? That is my question. So I think ideally I would say, okay, I have a positive assay, I classify, and then I don't care if I have negative assays. Because then I have you say, okay, this battery has measured whatever it has measured, and I have something positive. I go with that. Yes, I may run the risk of missing something because the battery is not complete, but that's a different question. So let's just focus on this, because now, and then I get a bit worried when I start reading the draft proposal for the pesticides. That essentially just says, okay, we have a positive. Now you do a lot of stuff and dribble away the results, because that's essentially what it says. Do a weight of evidence and then forget about it. And that's not really protecting human health. So in order to get it fully usable, I would like to see some kind of link to regulatory requirements also of what to do next, because otherwise we are just going to be stuck with these positive results, and as a regulator not know what to do about it other than requiring a DNT study, because that's the only thing we can do at the moment.
So
do you see the challenge?
So thank you for this great question. And luckily, we have the OECD DNT in vitro battery expert group, because this is exactly what we've been discussing in the last expert group meetings. And there will be a workshop report coming out very shortly. So within the next weeks, it's basically done. And there is one chapter on a possible tier testing approach in there, which also puts this battery as a tier 1 into context. So starting with some information on maybe also structure-activity relationships, then doing the DNT in vitro testing battery. If there is a positive fit, I think you have to put it in some sort of AOP context and to understand what it really means, and maybe even do a hit follow-up. For example, if you have just one hit in one of the assays, this might be of very high concern, because if only your proliferation, for example, is inhibited, or only your synaptogenesis, you can expect an adverse outcome. But, so a hit follow-up might be necessary. Then the question is, what would be the hit follow-up? And there are different possibilities out there. So this has not been really put into place, but the OECD is trying to also come up with guidance now, how to place the battery in a context, because you're right. I mean, if you're just stuck with one concentration response curve, so what shall you do? Plus, there are training webinars that we have been recording across experts from the whole group that will be available, that also help, I think, with how to handle this battery. I hope I answered your question.
Okay, thank you very much. And I think David Bell has his hand raised. Please, David.
Okay, I just noticed that the in the invitation for this, one of the things that you're coming to is how ANT-DNT NAMs could potentially fit into the future CLP paradigm for neurotoxicity. And I think I've got two levels of things which leave me maybe a little bit confused. One is the distinction between the classification criteria for adult and for developmental neurotoxicity. In adult, it's based effectively at the moment based on the concentration at which these things cause severe adverse effects, whereas in developmental, it's more of an all or nothing basis. And I don't really see that reflected in the considerations today. They're very different paradigms for coming to it. The other thing is, because it was how these NAMs could fit into the current or future CLP paradigm, again, I'm a little bit confused, because part of the time I'm hearing that these things can be definitive measurements of neurotoxicity, which can result in the most severe regulatory action. On the other hand, I'm hearing, and maybe this is just as I haven't followed it correctly, that they're a basis for concern. And this is all without delving into the detail of what is actually a positive in the battery. I appreciate there's always a little bit of there's always discussion of the specific details of studies that result in classification for developmental neurotox. But I mean, one of the things you were going to cover is how these are going to fit into the future CLP paradigm for neurotoxicity. So are there thoughts that you have on this?
Maybe I can start. So you mentioned that the current ANT or neurotox regulation is based on the magnitude of effects, and I think this can be really easily adapted by the in vitro. And so there has been the NAM Designathon, which has really tried to address this, and we've been also for the neurotox trying to, you can calculate from the doses you dose an animal to actually concentrations that we dose the cells with in vitro and have the same low, middle, high dose. And then, you know, for the DNT, I mean, I agree that it should be not a dose dependent, but a yes or no question that you can do with the NAMs. So this would be my answer, but maybe Tamara or Johnny, you have also some additions to that.
I mean, my point would maybe I mean, maybe it was confusing from the angle. I think the CLP and how this fits our story, maybe it's a bit vice versa, because there is a separation in the current CLP with the DNT and the ANT, and kind of the question is if this makes sense to have one within STOT and the DNT placed somewhere else. And this is kind of how the idea of this project came up. So it's not like and ideally in the end, we have an idea how to direct it into a CLP-like framework, but it was kind of the other way around. We were not like, we have an idea how this fits into a new CLP class. Let's run this idea of a project. It was rather like the current situation separates it so clearly that this hypothesis, if we not could look into this with NUMs, came up. So it was a bit the other way around. And yeah, ideally, we have an idea in the end how this could look like, but it will be different, or at least that's my feeling. I mean, we cannot replay or recapitulate an animal test and brain weight, and you know, we have to rethink it a bit, of course, because the approach will be a bit different from what is used now. I don't know if we can then say this is .SE1, this is .SE2, this is .SE3. It will be different. But, so that's a bit my point here.
And also for this question, we need the collaboration with ECHA. I mean, clearly, academic scientists cannot solve this issue on their own, and this is why we really appreciate the exchange also that we had with Kai at the recent Neurotox annual meeting. So we need to find a solution together.
So that's very kind. You've addressed the question I put about adult versus developmental for classification, but on the issue of DNT classification, I think I've heard at various times that the in vitro NAM battery is definitive, and actually it's a basis for concern and further work. Which is it?
So I think you're maybe referring to what I said. So I think the idea, when is the battery quote unquote good enough? And I think now there's more and more studies showing that the battery as it is now with the 17 assays is a really great starting point and can be used to make decisions. But in that OECD 377 review, that came from you all, that came from ECHA and from EFSA and other reviewers that highlighted it, that the battery should evolve with the science. Yeah, so it's a really difficult question to say when is this battery completely done and complete, and to do that, I think we have a really good shot of delivering information on that through this PARC 5.2.1 project, because we have a 96 chemical test list. We're going to be evaluating it. It has matched data within the DNT IVB that's already been collected. And then we look at these next generation NAMs and we see, can any of them add value or not? And if they can't, they're not necessary. But if they can, then you could potentially add in additional assays to improve predictivity. So this is a question that we're actively engaged in, but it shouldn't prevent using this great tool that we have already.
Yes, thank you very much. We are running behind schedule, so I suggest we stop now, but it was really an interesting discussion. I hope it has been helpful for you to build your PARC project. And in any case, ECHA is involved in PARC and is following your project, so we will for sure continue the discussion. So I would like to thank you for the nice presentation. Thank you to everyone also for the nice discussion. And let's continue the discussion on the PARC. I wish you all a good afternoon and talk to you soon.
Thank you very much.
Thank you.
Yeah.